My name is Mariana Wolfner. I'm the president of the GSA, and I'm here today with Elaine Ostrander, welcoming you back to another lecture in our 2024 GSA Awards series. So this past March at the TAGC meeting, Elaine received the Ed Novitski Prize, which recognizes scientific experimental work that solves a difficult genetic problem. It was established in the memory of Ed Novitski, a Drosophila geneticist and longtime GSA member, to recognize those who tackle scientific issues with creativity and intellectual ingenuity. Elaine was recognized for her work developing the domestic dog as an experimental system to solve fundamental biological problems and identify genetic sequences of relevance to human health and disease. Including work on human disease and behavioral health, she's demonstrated a dedication to creative methods for understanding canine genetics and the value of translating research organisms to human genetics. Elaine is Chief and Distinguished Senior Investigator of the Cancer Genetics and Comparative Genomics Branch at the National Human Genome Research Institute of the NIH. She also heads the section of comparative genetics and has published more than 375 papers. She's won multiple awards, including the American Cancer Society Junior Faculty Award, Burroughs Wellcome Award for Functional Genomics, the Asa Mays Award, Lifetime Achievement Awards, and in 2013, she received the GSA Medal for her work. She's also earned multiple mentorship awards and is an elected fellow of the AAAS and the National Academy of Sciences. The Ostrander Lab works in both human and canine genetics using state-of-the-art genomic approaches to identify prostate cancer susceptibility genes in high-risk families and in population-based case control studies. Her lab was one of the first to describe a role for the BRCA1 and BRCA2 mutations in women drawn from the general population, revealing epidemiological features for women at risk for breast and ovarian cancer. Today, she'll share more about her lab studies of the domestic dog as a well-phenotyped species with an extensively documented population structure that allows for solving fundamental biological problems. The lessons from studies of the domestic dog, can be applied to tackle problems in cell and organismal biology as well as human disease. Towards this end, the Ostrander Lab assembled the first catalogs of canine genomic variation, built the first linkage map, first described megabase linkage disequilibrium in dogs, showing how large loci could be reduced to single variants by taking advantage of breed relatedness. They continue to build a phylogeny of breeds and expand studies to learn about disease susceptibility, working with collaborators to identify genes for Addison's disease, retinopathies, and other diseases, including cancer. Before we hear from Elaine, I have two quick housekeeping items. The first is that all GSA online events, including this one, are covered by a code of conduct, and the link for that is available in the chat. The second is to ask you to put any questions for Elaine in the chat, and we'll get to as many as possible in the Q&A portion at the end of her presentation. So with that, I'm just delighted to hand it over to Elaine to tell us about her inspiring work.
Oh, thank you so much. Wow, that's some introduction. That's very gracious, and I certainly appreciate it. So I'm really talking today on behalf of just an extraordinary group of graduate students and postdocs and trainees, as well as a huge number of very valued collaborators. And I'll try and mention names as I go along, but please forgive me if I don't. That alone would probably fill up all of our time. So I'm going to talk about dogs and their genes, and I'm going to span a number of topics. This is one of those seminars that's got sort of something for everybody. So let's go ahead and dig in. Okay, why are dogs amenable to genetic studies? Why did I decide to, as somebody once said to me, do this to my career? Well, dogs exist in very large numbers. There's nearly 90 million owned in the US. They live in our environment. They're exposed to the same things as we are. They get the same diseases that we do, and they receive extensive preventative and diagnostic health care, probably more so than any other animal, with the possible exception, cats may be right up there too. But in 2022, Americans spent $137 billion on their pets. And about half of that was on healthcare, medicine, vet visits, insurance. But a lot of it was also on things like training and grooming and all those other things. In dogs, we can often sample multiple generations due to the very short lifespan that dogs have compared to humans. And that makes canine genetics in some ways much easier than human genetics. You do a study in humans, you may only get a couple of generations of DNA. You do a study in dogs, and you can get 5 or 6, or as long as your career goes. And so that makes them, really terrific for doing genetic studies. Now, my lab studies a lot of different things. We're interested in dog history, the lineages of how different breeds develop. We certainly study cancer susceptibility. This is a Bernese Mountain Dog. And about 25% of those dogs will go on to get histiocytic sarcoma. And those that do all will die of that disease. And you can see there's a locus circled on canine chromosome 11 that we were the first to describe that increases susceptibility for that disease. We study breed organization, how breeds relate one to another, and how we can take advantage of that fact. I'll tell you more about that. We also study morphology, and I'll talk about that as well. Body size, as well as ancient alleles. So where do all the differences in morphology between breeds come from? And we'll talk about behavior. So those are our 4 topics for today. I will say we have studies going on all over the world, including in the Galapagos Islands. We've been very fortunate to get a large set of samples from Chernobyl. We're looking at what changes have allowed those dogs to live and stay and breathe in that environment. And the Dog 10K project that I had, which is sequencing thousands of dog genomes and putting them out there in the public for everybody to use. Okay, dogs. Well, they were domesticated just an evolutionary drop in the bucket ago. So 20, 30,000 years, likely in Northern Europe. Dogs with jobs. So if we think of livestock guardians or sled dogs, that's something that happened about 10,000 years ago. But breeding of dogs in earnest to be able to do certain things that were important for our survival began about 5,000 years ago. But most dogs, most breeds that you see running around the dog park have only been around for about 200 years. And there was an explosion of breed formation during the Victorian times, largely led by people in Western Europe, fanciers, breeders, people interested in producing a breed that was different than this, but more like that. So most of what you see hasn't really been around for a very long time. Now, modern dog breeds display an amazing diversity of traits in terms of their morphology, their behavior, as well as their disease susceptibility. So you see examples of all of that on the 4 pictures shown. The largest to smallest breeds of dog differing by about 40-fold. You see a border collie herding sheep. You just see some of the changes or variation that exists in dog morphology and how that makes us think of humans. So some of the same underlying genes clearly at work. And these are melanoma slides. It's just an example of the fact that dogs get the same diseases we do, are often treated the same way, respond to treatments in similar ways. And so they're a really good place to study the genetic susceptibility of disease. Things to remember during the talk: dogs are the most morphologically diverse land mammal on Earth. All dogs are members of the same species, Canis lupus familiaris, and there are about 350 recognized or registered breeds of dog in the world, but there are also lots of other defined populations in special geographic areas. And my lab is in a long-term partnership with owners, with breeders, with veterinarians, with dog lovers to collect DNA samples and some information, mostly about pet dogs. We don't breed any dogs. We don't keep any dogs in kennels. We don't have any dogs here at NIH, any of those things. This is a citizen science project that's allowed us to collect nearly 40,000 DNA samples in the last 30 years. All right. So, where are we collecting dogs from in order to address all those questions? Well, everywhere. Everywhere that we can get in, we're collecting dogs from. And this is an outdated map, so there's lots of additional places where we have collaborators or colleagues on the ground who are sampling really interesting data sets of dogs with interesting traits and sending them to us. In Papua New Guinea, we recently showed that the New Guinea singing dog was not extinct in the wild, for instance. It actually does still exist. In Chernobyl, as I said, we're looking at dogs living in and around the power plant. Um, in Nepal, we're looking at dogs living at different altitudes to see what genomic adaptations have occurred. In Greenland, we're looking at the history of dog migration around the island because that's very likely to parallel human migration. Where dogs go, where humans go, they always bring their dogs. So lots of fun stuff if you're in my lab. Now, this is one of the most important experiments we've done. It's done by Heidi Parker, and we've updated it several times, including very recently. And now there's so much data on it. It's not we can't put it on a single slide. But what this is a neighbor joining dendrogram. So we've taken in this iteration 161 different dog breeds. We got about 10 dogs for each breed. And then we took the DNA from those dogs and we interrogated it at 170,000 different positions in the genome. And then we said to the computer, well, how do these dogs relate one to another? And what the computer said is, well, they divide into 23 clades or groups. And you can see those 23 clades are indicated by color coding. And for the most part, this makes pretty good sense. So up here are the terriers in aqua, the Yorkshire, the Silky, the Australian, the wire fox, the Jack Russell, etc. Lots and lots of terriers. Down here are the spaniels, English Springer, Cavalier King Charles, American Cocker, Field Spaniels, English Cocker Spaniels, and so on and so forth. And we ever add to that and get more and more and more resolution to understand how different dog breeds came about. So why is that so important? Well, we rely on it for every hypothesis we develop. So let's say I'm working in disease susceptibility. Let's say it's a common disease like let's pick epilepsy. And let's say a dog that walks into the vet's office is a Boston Terrier. Well, you can reasonably hypothesize that whatever the causative mutation is for that Boston Terrier, any epileptic dogs that are boxers, bulldogs, dog de Bordeaux, bull terriers, etc., they carry the same mutation for their disease because these guys share very recent common ancestors. And we can go over here and look at those terriers. And let's say a Scottish Terrier walked in, same situation. You could reasonably hypothesize that any affected Cairn, Norfolk, Norwich, Yorkshire, etc., Australian Terriers that came in with epilepsy likely share the same mutation because they have a recent common ancestor. What's going on here in the Terriers? Almost certainly going to be different than what's going on here in these bully breeds, because you can see that they're in all these a great distance away from one another in this circle. So we do this all the time, constantly, for everything we do. It tells us how to set up our experiments. Set up for students in the group that are listening is everything. You don't want to do the experiment over and over and over and over again. You want to spend a lot of time thinking about how you're going to set up your experiment. So let's talk about cancer. Many breeds are at an elevated risk for specific cancers, and we study those because we believe that can help us understand susceptibility to human cancers. So osteosarcoma is something we see in the long limb breeds like the Scottish Deerhounds, Irish Wolfhounds, Great Danes. Bladder cancer, which I'll tell you more about, is something we see in Scottish and West Highland White Terriers, which, by the way, are really close to each other on the phylogeny you just saw, as well as Shetland sheepdogs. Malignant histiocytosis. I told you we see that in Bernese mountain dogs. We also see it in flat-coated retrievers, where about 20% of them get the disease. Otherwise, it's a pretty rare cancer in dogs, and those two breeds are not themselves related at all. So we're probably going to find different genes as we investigate those. And then gastric cancer in the Belgian breeds, like the Belgian Tervuren or the Belgian sheepdog, and also, as I've shown for you here, in the Chow Chow. This is a bad one. In humans, it's almost always lethal, and in dogs, it's the same thing. So finding those genes is something that one of my postdocs, who now has her own lab at Cornell, is devoting all of her time to, Jacqueline Evans. So let's talk a little bit about invasive urinary cancer. It accounts for a small percentage of tumors in the dog, but because of the number of dogs, that translates to 50,000 newly affected dogs each year. 50% of the time it metastasizes, and then the average survival is less than a year. Treatment has not evolved in the last 50 years. We're just still doing the same old thing. Now, if you look at these three breeds, this is pretty striking. If you're a Scottish Terrier, your odds of getting invasive urinary cancer are 21-fold higher than the average mixed breed dog walking down the street. If you're an American Eskimo dog, it's 6.6-fold higher. Shetland Sheepdog, 6-fold higher, and a West Highland White Terrier, 5.8-fold higher. And these are all statistically significant. This is extraordinary. 21-fold higher, 6-fold higher. Clearly, there's underlying genetics here that we for sure want to tease apart. And in the study I'll tell you about, which was just published um, 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 in one of the major cancer journals, Precision Oncology, I'm going to tell you about our studies in the Shetland Sheepdog. And these dogs, by the way, belong to Dana Drager, one of the staff scientists in my lab, who is an aficionado of Shetland Sheepdogs and has always owned them and will always own them. Okay, so you're looking at a genome-wide association study. All the dogs are Shetland Sheepdogs in this study. Each one of these gray, black, indicates a different dog chromosome, going from chromosome 1 to 38, which is how many chromosomes dogs have, as well as the X. We compared the DNA from 49 cases and 50 controls, again at 170,000 positions in the genome. Now, this doesn't sound like a lot, but in the dog world, it's not the number of individuals you interrogate, it's the number of lineages. So none of our cases share common parents, share common grandparents, share common great-grandparents. And we can actually cover a big chunk of the breed, in some cases the entire breed, with really modest numbers of samples. So when we did this, we found 3 markers, single nucleotide polymorphisms, we like to call them SNPs, on canine chromosome 13. And you can see anything above the line is statistically significant. And you can see we have all these dots here above the line, telling us there is something on chromosome 13 that is important in risk in the Shetland Sheepdog. We did all the appropriate corrections for population structure and significance and permutations, and the result held up really well. Now, remember I told you that our story gets much better if we can combine data from related breeds? Well, this is a perfect example of that. So if you look over to the right, when we looked at Shetland Sheepdogs, SSD only, we got a p-value of 3 × 10⁻⁷. But I went to my wheel and I said, what can I combine with my Shetland Sheepdogs? So I combined affected collies, affected Australian Shepherds, and affected border collies. These are all herding dogs. And now my p-value went up to 5 × 10⁻⁻10. So when you look down here, you can see that line, those dots, those red dots go up way further, lots of statistical significance, and for sure there's something there. So we've done a lot of work on this, and we've done a variety of different statistical tests and different kinds of GWAS, and here's what we've learned. We've learned that there's a gene called NEPAL1 that has a mutation. And that mutation is really important, and it really increases risk of getting the disease in the Shetland Sheepdog. That mutation is also overexpressed in canine bladder cancer cell lines. And if you're a homozygote, you have 2 copies versus 1, your risk goes up even more. But there's also other genes that modify risk up or down. There's one on canine chromosome 28, and at that position, you can have a C or a T. There's others on canine chromosomes 4 and 9 and 21. And when you put this all together into a picture, you begin to understand, oh, this is why cancer doesn't make sense a lot of the time. Or 3 people in my family have this cancer, but my brother and I didn't get it. How can that be? Well, it's because all of these things contribute. And in this case, we're talking about 5 different genes. So Nepal1 increases or doesn't increase your risk depending on which alleles you have. If you're a heterozygote, it's going to be in the middle. If you have the minor allele at chromosome 21 or 28, it continues. If you have the minor allele at chromosome 28, you get a no. If you have the other one, you get a yes. And if it goes, if you're a homozygote for the GG at Nepal1, then and you have the major allele at chromosome 21, it goes yes, et cetera, et cetera. And for our discussion, the details don't matter. But the point is that these different combinations of variants at these 5 loci, these 5 genes, can either increase your risk, they can decrease your risk, give you a protective effect, or not change your risk at all. And so that's why when we look at families, sometimes we're just completely confounded. And it's because there's multiple genes acting at once. But dogs taking advantage of breed structure are a great way to sort that out that we really just don't have in humans. And so we're doing that for lots and lots of different diseases now. Now, using those same principles, we've been enamored of morphology in dogs for about 15 years now. And this picture just shows you some of the differences we see across breeds in size and leg length and skull shape and coat color and coat texture and everything that you can possibly think of. So we've been studying body size for quite a while, and we've identified a number of genes, which in aggregate probably account for 85 or 95% of the variation we see in dogs, particularly in the small to medium-sized dogs. Now, that same experiment in humans identifies hundreds of regions of the genome, thousands, in fact, now. So thousands of different places in the human genome have variation that contributes to human height and human weight. Whereas in dogs, we have these much more manageable numbers, and that speaks to the evolutionary structure. Dogs have only been domesticated for 30,000 years, selective pressures in the last several thousand years by humans, and then the strong breeding for traits a couple hundred years ago. So a reoccurring theme in dog genetics is that we see small numbers of loci that have a very large effect for given traits in dogs, whereas it's the reverse in humans, where that same trait will have lots of genes, lots of places in the genome that contribute, but each one contributes just a tiny bit. So given my choice, I'm going to study these things in dogs. And that's exactly what we've been doing. Now, I could give this whole seminar on dog breed body size because I just love the topic, and I love looking at ancient DNA and figuring out where these variants came from, what part of the world, and what kind of wolves 50,000 years ago. And that's some lovely work that Joss Blase in the lab has done. But everybody loves dog behavior, and everyone who has a dog has strong feelings about dog behavior. So I thought I'd spend the second half of the talk talking exactly about that. So different dog breeds, different behaviors, herding dogs, sled dogs, sighthounds, retrievers, draft dogs, pointers. So we've been interested in doing genetic studies of these stereotypic breed behaviors. Now remember, breeds initially resulted from a human need for a specific behavior that was really critical for survival. When we talk about an instinct, we're talking about behaviors that occur early in life on the occurrence of a specific triggering stimuli, like a bunch of sheep walking by a border collie, and naturally without prior training. And again, a bunch of sheep walking by a border collie. They really know what to do. So Emily Dutro in my lab just had a beautiful paper come out in Cell about a year ago, and she looked at herding lineage. So the dogs that it would have been in that clade or in that cluster, and then she showed further what the lineage of those dogs was, what were the earliest, and what they evolved to, and then what we have that are the most diverse today. And what she found is that they share an extraordinary number of genes. They're each colored here in green. And these are all genes that are in axon guidance pathway. So if we're going to look at herding dogs and we're going to say, what are possible things that could be important in having herding dogs herd? Well, we would say that these are things in the axon guidance pathway. And in fact, she showed that these are genes that develop very early on embryonically in the brain and are obviously very important for a variety of things. This includes axon outgrowth genes, axon attraction genes, and axon repulsion genes. Now, if you want to speculate as to how dogs actually herd sheep, in the context of these genes, we have a hypothesis. We haven't proven it yet, but we do have one. Many of the genes that she highlighted were part of the ephrin signaling pathway, like ephrin A5, ephrin A3. So how can something like the ephrin A5 contribute to herding? Well, EPHA5 and its ligand have been implicated in anxiety and in maternal pup gathering in mouse models. So the mouse mother will try and keep all of her babies together, next to her, all kind of herded up together, herding being the operative word. So we speculate, and this is, how science is driven by speculation, by forming hypotheses, by doing experiments. Herding drive could be, or it could involve augmentation of the same anxiety associated pathways that drive maternal protective behaviors. And that's going to be fun, really fun for us to look into more based on this work of Emily Dutros. So I want to just tell you one more thing, 1.5 more things about behaviors. And that is a really cool study that Jae-Min Kim did. He also has his own lab now. And Jae-Min got the idea to sequence lots of dogs that were sport hunting breeds. So these are breeds that assist hunters by retrieving, flushing, pointing. It requires prolonged periods of physical activity. And he compared that to lots of sequences from terrier breeds. So terriers were originally developed to rid land and buildings of vermin. They require short, rapid bursts of activity. So these are both very athletic. By the way, this paper appeared on the cover of PNAS. Both very athletic groups of dogs, but they execute what they have to do in very different ways. And he wanted to see at the genomic level what was going on. So he looked for signatures of selection. And when he did that, he found that the signatures he found fell into 3 groups. They were important in neurologic function, for instance, in learning. Some of the genes that he identified when mutated in humans were associated with learning disabilities. They were important in muscle function, muscle contraction, muscle development, fatigue-enhanced muscle pain. When mutated, we would see muscular dystrophy in humans. They're associated with muscle mass. And cardiovascular function like heart rate and hypertension and smooth muscle contraction. And perhaps we shouldn't have been surprised because we know from other writings that elite athletic performance is largely determined by the integrative roles of muscle, cardiovascular, and neurologic functions. And that is exactly what he found. So let me tell you about 2, MST1 and TRPM3. So these are genes that are really important in racing performance. And we've looked at this in whippets. And so if you have the right alleles for these 2, you will race 27% faster than whippets who do not have them. And this whippet, for instance, is a heterozygote for the myostatin mutation. And this mutation completely knocks out the gene. And you can see it's a beautiful dog. Doesn't look like that standard, very thin whippet, but has more like a racehorse look with all the musculature defined. This is a homozygote whippet for the myostatin mutation, and that's kind of overly muscled. So that was pretty cool. And we dug on. So the next thing we looked at was ROBO1. We know that's important in neurologic function. And this turned out to be a biggie if you're looking at something called agility. So agility is just course racing. You jump, you go through tunnels, you go in and out stuff, you go over stuff. you can look at how good a dog is at agility by looking at their number of agility titles 1 per breed or per dog divided by number of dogs registered in a geographic region or by breed as a whole. And what we found was that ROBA1 variants were very significantly associated with how well dogs did at agility. So ROBA1 plays a role in regulation of neuronal migration, development, and axon guidance and growth. In humans, when it's mutated, it's associated with a developmental dyslexia. It's a learning disability associated with an inability to translate visual cues. So in dogs, ROBA1 seems to affect the ability to identify and learn the environmental information that's telling them what to do when they come up to a particular obstacle in their obstacle course. So think of American Ninja Warrior gone completely wrong, and that would tell you what happens if Robo is missing. Okay, so I think that's what I'm going to say about behavior. But this is definitely the part of the seminar where everybody raises their hand and says to me, well, it's great that you're spending all this time studying purebred dogs, but most of us have mixed breed dogs. So what about mixed breed dogs, or some people would say mutts? I don't know. Maybe that's not a very nice term. Don't you ever collect them? Don't you ever include them in your studies? And the answer is increasingly yes. But like everything else, you have to develop your hypothesis correctly. So we started out our thoughts about mixed breed dogs by looking at designer breeds. And we looked at the Labradoodle, the cross of the Labrador Retriever and the Poodle, first done in 1989. It was actually done in the first one, I think, was done in Hawaii. The idea was to create a service dog that was allergy and asthma friendly and had low shedding. That, of course, coming from the poodle, but they wanted to be a service dog, a friendly dog, a trainable dog, and that was coming from the Labrador Retriever. So we entered a whole bunch of Australian Labradoodles in our study, and to be eligible, you had to descend from 5 or more generations of crosses of Australian Labradoodle to Australian Labradoodle. So the dogs that we had were pure Australian Labradoodle. And we then went back and we looked at first generation crosses, second generation crosses, third, et cetera, from going from generations 4 all the way to 8. And a lot of the first generation crosses actually were hard to be able to look at, but we had their pedigrees, and so we were able to pick them out. Excuse me. So this was a really fun study, actually, and I had no idea what we were going to find. And I just wanted to know what an Australian Labradoodle was, right? To be one, you have to come from 5 generations of Labradoodles. But when we look at these generations, what is it we see? What are we finding today? Well, this was a huge surprise. So this, again, is chromosome 1 through 38 and the X. But now all we're doing is looking at allele frequency. So you compare poodles to Labradors. They have very different genomes, and everywhere there's a significant difference. You get a red dot, you get lots and lots and lots of red dots on every chromosome, and that's what you would expect. If you compare a Labrador Retriever to a Labradoodle, you get a lot of red dots, but not as many. That makes sense because the Labradoodle is part Labrador Retriever. But look what happens when you compare a poodle to a Labradoodle. You get almost nothing, telling you that your Labradoodle is actually mostly poodle. And that's what breeders have been selecting on to get dogs with traits they want. They turn out to be traits that poodles have. Poodles are smart. Poodles are so, quote, hyperallergenic. Poodles are great dogs. And so for sure they're getting some Labrador Retriever traits, but since the things they care about the most have to do with the coat, and trainability, most of what is being selected is poodle stuff. So when you compare poodles and Labradoodles, they're essentially getting the same dog. So that was really interesting to us, and it told us that the word mixed breed or mutt didn't really mean anything. And when we look in the literature, you start looking at mixed breed dogs, and you find that in a particular kind of mixed breed dog, you may find a disease that's typically only found in one or the other. So congenital ichthyosis mutations found in golden retriever poodle cross, but that's not something both breeds typically get. Exercise-induced collapse we see in Labrador retrievers, but in other mixed breed dogs. Same with diabetes, same with various ophthalmal vision disorders, ophthalmic abnormalities. And so we did a little experiment, and what we find is how healthy your mixed breed dog is depends on the health of the parents. So here's a collie. Collies are sensitive to drugs. You can't give them heartworm for the most part. They have a mutation in a gene called MDR1. 69% of collies have at least one copy. So now let's start looking at some mixed breed dogs. Well, this is Grace. She doesn't look like a collie, but we actually know her lineage because a friend of someone in the lab bred her. And Grace is 12% collie. So she does have a risk for drug sensitivity to MDR1 because of MDR1, but it's a pretty low risk. This is Sophie. She's 73% collie, so she has a very high risk of drug sensitivity. So knowing the component breeds of your mixed breed dog turns out to be really, really important in thinking about their health. As a geneticist, it becomes very important and very useful for me to include these dogs in my genetic studies because Grace only has 12% of the collie genome. So if I'm looking for a collie trait, I can look in that 12% of her genome if I can identify it, which we now have approaches that can. Here's another example. This is an American Staffordshire Terrier. 26% of those are affected by hip dysplasia, and a French Bulldog where it's 33%. This is Hattie. She's 25% American Staffordshire Terrier and 12.5% French Bulldog. So Hattie's at a pretty high, pretty moderate, I guess you might say, risk of hip dysplasia. So she's a dog you would want to really think about the food she gets. You would want to make sure she's well exercised, that she doesn't get overweight, that she gets her hips certified before breeding, and all those sorts of important things. And so this is my last slide. There are 152 disease-causing gene variants that have been identified in dogs. Most were originally described in purebred dogs, and they're kept track of in a database called OMIA. You can go and look up diseases or traits or your favorite gene and see what the relationship is. But a really interesting report came out from Hannes Loewy's group in 2018, and it analyzed 100,000 dogs. And they found 80 variants were seen in purebred and mixed breed dogs of these 152. But they found 34 variants were seen only in mixed breed dogs, only. So genetic testing allows breeders to eliminate disease variants in purebred dogs, but crossbreeding has allowed variants to remain hidden in the dog genome pool, and they're hiding in mixed breed dogs. So taking your mixed breed dog, depending on what you cross it to, you could suddenly see things that you haven't seen in your dog's lineage or whoever you got the dog from ever, or certainly in a long time. So Benji is 17% Labrador Retriever. He's at risk for exercise-induced collapse. Bogey is 27% Australian Shepherd. He's at risk for heartworm medication complications. That's that MDR1 gene. And you already met Grace and you met Sophie. And here's Cindy. She's 15% poodle. Well, the owners might want to, again, be really careful with diet, thinking about if she's at risk, low risk, albeit, but low risk for getting diabetes. So that's my speed talk. I've tried to convince you that dogs are a great system to study the genetics of morphology and disease and behavior because of variation that's trapped in their genome. Most traits studied thus far are controlled by small numbers of genes of large effect, and studies of breed-enriched canine cancers identifies risk alleles that are really important for understanding our own cancers. And the dogs have been selected for certain behaviors necessary for survival. And understanding those genetic underpinnings certainly informs studies of human behavior and human biology. So here's a list of collaborators, just amazing group of people. And I'm going to leave that up and take some questions.
Thank you very much, Elaine. That was a really, really cool seminar and really interesting, all of it. I'm gonna now have some questions, and I'm gonna start off with an anonymous question that came in through the chat. And after that, we have questions in the chat. Will go in this order: Sylvan Urfer, Christopher Jones, Wyatt Toure, and Sean Beckwith. After that, if we still have time, you can either post them in the chat or raise your hand and we'll call on you. So the anonymous question was fairly early in your talk when you were talking about the GWAS. Said that it appears that your GWAS significance bar is lower than what's used typically for human GWAS. Is this because you're using fewer SNPs because haplotype blocks are longer in dog? And if so, do longer haplotype blocks confound fine mapping and risk allele identification?
Oh, indeed they do. So you're absolutely right. LD blocks are longer in dogs than they are in humans. And so when we used to just use 170,000 points or markers, we would get a block or a haplotype that would be 2 megabases in length. And there could be 40 genes in there. And then we would have to really weed through. And the problem that came about in doing that is people would kind of pick their favorite genes. They'd say, oh, I'm studying behavior and I have this 2 megabase haplotype block. And this gene is important in something in corpus callosum something or other. So I'm gonna bet it's this gene. And of course that's not the right way to do science. So now we sequence everything. And so that's what the Dog 10K project has been. And we just put out a paper with 2,000 dog sequences spanning, I think, 300 breeds and populations. All the data is freely available. We made it available before we published. We put out the pipelines. We put out, all the files, all the variants, all the copy number information, the mitochondrial information, so that people don't have to do that anymore. But you're absolutely right. If you're just using a set of markers and you're looking at LD blocks, it gets really hard to go from linked marker to gene, for sure.
Thank you. Sylvan, would you like to ask your question?
Sure. Hey, Elaine.
Hi, Sylvan. How are you?
Pretty good. How about you? I was going to ask a question from the behavioral sciences. So there's a theory that most breed-specific behaviors are really part of a truncated predation behavior chain. So you have like herding dogs that just stare at other animals, or you have pointing dogs that stop and point at their prey. And I was wondering if you could comment on that theory from a genetic point of view.
Yeah. So just to rephrase this, so if you think about border collies and they have this unwavering stare that kind of makes the sheep move away, that they use to control direction, or they have this crouch where they drag their bodies along the ground like they're about to pounce, that reminds you of some other, like a wolf or something. I don't know my wolves very well, but going along the ground and getting ready to pounce. And so are things like crouch and stare and border collies sort of a holding back of those? Are they those same genes, but there have been different alleles that have been developed that kind of hold those things back or control them? I don't know. And I think certainly our data doesn't yet show that for sure. I don't think, we don't have any functional data or validation for the genes that we found, for instance, in herding dogs or in other aspects of herding that Dana Drager is studying, that really says this is it, this is it, this is it. We're really looking at differences between, for instance, herding and non-herding populations. Other people will argue yes. I think people who are true behaviorists will argue yes. And has there been a marriage of genetic, functional, and behavioral data yet? I don't think so. Not a very tight one. I think there's been, dating, but I don't think all those things have quite come together. So it'll be really interesting to see, for sure. And one would hypothesize, that's a great hypothesis, but I think we're a little bit short of it still at the genetic level.
Right. Thank you. Appreciate it.
Thank you. Christopher Jones, would you like to ask your question, please?
Yes, and I remember to put the audio on. Thank you. Yeah, so I was really interested to see the differences that you showed between poodles, labradoodles, and lab.
Yeah.
And I noticed that it looked like there were many more differences between labs and poodles. I'm guessing, but I just wanted confirmation that because of the restricted parentage,
Yep.
the differences between labradoodles and labs and poodles and labradoodles adds up to less than the differences that you showed between random labs and poodles.
You know, I didn't actually calculate that for sure, but poodles and labs are not closely related at all. And so for sure, we're going to see a ton there. So is what we see between labs and labradoodles plus poodles and labradoodles equal what we saw above?
Right.
I'm
It
going to
looks
guess
like
not.
no.
I'm going to guess not. Yeah, I don't think they would at all. And I think that has just to do with, you know, population structure and sample sizes and, you know, all those sorts of things. It's actually a really good question. I should go and think about that some more.
Thank you.
Thank you. Next question, still about labradoodles, Wyatt Touré.
Hi, yeah, great talk. I was just curious, I might have missed it, what generation of Labradoodle was used in the comparison that shows that Labradoodles are mostly poodle. And then I was curious about of the Labrador, variants that stick around, what are those variants producing? Like what Labrador
Sure.
traits do people either knowingly or unknowingly keep around?
Yeah. So I was stumbling a little bit when I told you that. So to be a registered Labradoodle, you have to have come from 5 generations of Labradoodle, right? Your parents had to be Labradoodles, your grandparents had to be Labradoodles, your great grandparents had to be Labradoodles. So I wasn't being terribly articulate at that point. Our experiment involved multiple generations of Labradoodles. So, you know, I think we started with 2nd or 3rd generation Labradoodles and 4th and 5th and 6th, and we went down to 8. So we had all different generations of Labradoodles in our study. But to be a registered Labradoodle, you have to have 5 generations behind you of Labradoodles. So what we found was that the genes that are being selected for to make a Labradoodle are the same things that they relate back to exactly what the breed or Labradoodle was created for. And there are things that have to do with fur. They have to do with the undercoat. They have to do with fur length. They have to do with growth patterns of the fur. Because remember, the key here is they wanted a service dog that was trainable, that was approachable, that could function in crowds. But it had to be something that didn't start asthma attacks, that wasn't super allergenic, that everybody who got around one had an allergy attack. And poodles are really great for that. I mean, so are Portuguese water dogs and Irish water spaniels and others. And so it's not uncommon to breed poodles into lots and lots of other breeds in order to get something that's less, what people would say, less allergenic. Right. I'm not going to say it's hypoallergenic because there's no thing as a pure hypoallergenic dog, but to get dogs that elicit less of an allergenic effect. So this was graduate student's work, Mohammed Basil Ali. And the thing I really wanted him to do was I wanted him to go out and get all the other oodles, right? I wanted him to get dachshund oodles and Bichon Frise oodles. And, you know, because there's all, you know, if you look online, you go oodles, you just find oodles of oodles, right? They've bred them to everything. And I wanted him to go through and look at a bunch of those and see if they were also preliminary or primarily poodle. But, you know, he had to graduate. His committee said it was time. So we didn't get that experiment done, but we'll get it done someday.
Thank you.
There's
And
Charles Laird, an old friend. Hi, Charles.
So we have not a lot of time, and your talk has been so interesting and exciting that there are a ton of questions. So I think we'll just go through them in order. And
Okay.
I apologize to people who may not be able to get their questions asked. But the next questioner is Sean Beckwith.
Hi, Sean.
Hi, thank you for such a fascinating talk. My question is that given how quickly dogs have evolved under human breeding, if you find mutations associated with phenotypes that are due to transposons, would you only look for SNPs? Because I can imagine large
No.
changes to the genome
Yeah.
would come from.
That's a great question. So, you know, one of the most interesting studies that's been done along these lines have been done by Bridget von Holt of Princeton, and she's been looking at sociability behavior. And what she's reported is transposons that affect expression of genes in a region of the comparative region in humans is called the Williams syndrome region. And this is a region of multiple genes that when it's knocked out in humans, you have kids who have developmental delays, but they're hypersocial, right? And so when they compare wolves and domestic dogs, they see transposon changes in this region. And so the thinking is going from wolf to dog, transposon mutability or changes in this region was something that was really important in making that transition. So for sure, people are looking at transposons in lots of different contexts, but that's the one that people always refer to as a starting place to start thinking about it.
Thanks.
Thank you. Nelson Lau.
Thank you, Dr. Ostrander. That was a really fantastic talk. And one question I had that actually could relate to our general public is that there's a lot of activists right now frequently trying to put state legislation, like in New Hampshire and Europe, to ban the breeding of brachiocephalic dogs, like
Yeah, right.
bulldogs. And they say that they should ban it because it's a birth defect.
Yep.
What do you think about brachiocephaly? Is it a birth defect or is it more of a stable trait that's been bred in these dogs? Because that's what I provided testimony to New Hampshire legislature saying as a geneticist that these are stable traits. What should the public know about
Right.
brachiocephaly?
So brachycephaly is that sort of pushed in face that you see in things like bulldogs or French bulldogs, as opposed to the other extreme would be dolichocephalic. So that elongated nose and face that you see in something like an Afghan hound. So the problem with brachycephalic dogs is they have breathing problems for sure. And they're not small. I mean, they are significant breathing problems. So you're exactly right. I know of some place in Europe, and I'm trying to remember where it is that I just read, just banned
Germany.
breeding.
Germany wants to read.
Germany wants to, but somebody just did ban them. I think the responsibility actually falls to the breed club. And I've seen other breed clubs when they really became aware that a health problem was starting to dominate their breed, work with geneticists, find the underlying genes. And I know at least one of them has been found for brachycephaly. Actually, two have been found and published. And then, you know, develop genetic tests and work to breed healthier dogs. The Bedlington Terrier copper toxicosis story is classic. They found the mutation for copper toxicosis. A third of dogs were carrier. Dogs would build up, homozygotes would build up copper in their liver. It was lethal. The community begged geneticists to find the gene. They did. Genetic test is developed. So now you test your dogs before you set up your breedings, and incidence of the disease has gone way down. We did it for collie eye anomaly in collies several years ago. So I think the responsibility falls to the breed club to tackle this.
Is it a birth defect or is it a stable trait?
It's a trait, right? So I don't know what the I don't use the word birth defect, but I would say it's a trait that's under selection. That's how you want a bulldog to look. How stable it is in the genome. If you didn't breed the dogs with the breed standard to dogs with the breed standard to dogs with the breed standard, I don't know how quickly it would drift out. I honestly don't know. But what I do know is it's a problem, and it's up to the breed clubs to fix it. And I'm hearing that's what they're doing. I know I'm hearing a lot of encouraging things about that, because nobody wants miserable, short-lived dogs, right? That's to nobody's advantage.
Okay, thank you.
Yeah.
Thanks a lot. I think we have time for maybe two more questions, maybe three.
Sure.
So Anton Briantsev has a question.
Oh, I'm sorry. I didn't know that I meant to speak. Very interesting talk. I'm really sponge and everything. But I have a question. I work on Drosophila, and so those are incredibly resistant against inbreeding. So what's the story about the dogs? How resistant are they against inbreeding?
So what do you mean, how resistant are they?
I mean, you know, the gene, the sort of like when we do flies, there is a basically the term of isogenic line. So we know that for most part, all the genes would be in homozygosy. And what's the story with dogs?
Okay,
I mean,
so
male in general, I guess.
Yeah, I don't know enough about Drosophila to give a great answer.
How
But
homozygous are the dogs, basically?
right. So, you know, breeders recognize you do not want a bunch of homozygous individuals because lots and lots of diseases are going to, you know, come up. So, right. When we sequence, you know, our 2,000 dogs, I can't tell you how many millions and millions and millions of SNPs we came up with and copy number variants and et cetera and so on. So the goal has never been to homozygose the dogs. The goal has always been to get certain traits to breed true. So if, you know, part of the thing that defines a Dalmatian are N percentage of the body covered with very round spots that don't overlap, there'll be selection for that. But there's not going to be selection for a gazillion other things, right? And so I think breeders are more and more savvy now than when I started in the field, you know, 25, 30 years ago, about who you breed to who and being really careful not to breed individuals that are too close related. I think that was much, much more common, you know, 100 years ago. But when I go out and talk to breeders, they have a lot more knowledge than they did 30 years ago about partial penetrance and, you know, recessive traits and modifiers and epigenetic effects. And we talk a lot about aging because my lab has worked with Steve Horvitz on aging. And so and how does aging affect cancer risk? I mean, they ask all those really great questions. So I don't know that the comparison is really good. For sure, they have more homozygous regions than humans do. Absolutely. That's why we have those long stretches of LD, but nowhere near what you can do in Drosophila, and nobody wants to.
Thank you.
Thank you. I think one or two questions. So Anne Kwiatek.
Hi, Elaine. This was a really, really fun talk.
Thank you.
It's my favorite species as well. So I did have a question. You know, there's a lot of these direct-to-consumer genetic tests available for our pets. What do you think about those? Can you comment on accuracy?
Sure.
And so forth?
Yeah, I think they're great. I'm not allowed as a federal government employee to say this one's better than that one or that one's better than this one. But it's a general concept. Yes, I think it's great. I think if I had a mixed breed dog, I would want to know what the component breeds were, you know, because that would affect what I fed the dog, how I exercised the dog, its environment. You know, if I had a dog that was part border collie, I would not leave it alone in an apartment all day. It needs to go out and work at least an hour every day, right? So, you know, if I had a dog that was part greyhound, I would take it out on my bike and make it run alongside me so it could, you know, really burn some speed every day. So I think it's great. And I know increasingly these companies are offering tests to predict risk for certain diseases. I think in some cases that's based on breed, and in some cases it's based on knowing the actual associated disease allele or causative mutation. And, you know, for me, it would be important to know which of those things, you know, are you saying my dog has a 25% chance of getting blah because it's this percent this breed, or because it has this associated marker, or because, you know, you have actually found the mutation that's been reported. So I think there's a little bit of a blurring with some of the companies in that regard, and I would want to know before I made decisions. And I know that at least a couple of the companies will offer you kind of predictive testing. Like if you give them a DNA sample of your puppy, they'll say it's going to grow up to be this big and it's going to have this color coat and its ears are going to go up or ears are going to go down, which you really can predict because we found those genes. So that's legit. And, you know, I think that's fun and that's cool, actually. We've by no means found all the morphologic genes, so I would love to know how often they're right. I actually should ask a couple of them to give me data, and they've been actually really generous about sharing anonymized data. They get permission from the owner to share anonymized data, and then they do. And you know, we have people from some of these companies as authors on our paper because they share data to improve the health of dogs. So I'm enthusiastic about them for sure.
Excellent. Thank you.
With that, I just want to say thank you so much for this wonderful talk, and everybody for the wonderful question and answer period. And again, congratulations on this very well-deserved award.
Thank you so much. It's been a tremendous honor. And thank you all for listening to me. I hope it was fun.